Hello, and welcome to my biology role model project. My name is Sophia Reynolds, and I will be presenting on Hamilton Smith. Alright, so to give you a little overview, Hamilton was born in New York City, New York on the 23rd of August in 1931. Then, 6 years later, his family moved to Champaign-Urbana, Illinois. From there, his father became a professor at the University of Illinois, and his mother became a writer. So, based off of that, you could tell that they had pretty intellectual minds. His mother and father did, and they kind of influenced Hamilton and his brother to do the same, to kind of like look at life from the same perspective as them, which really introduced them into science, you could say, because they were given this little Gilbert chemistry set, and from there they played with it in the park and then just expanded on that knowledge and eventually started creating experiments in their home lab basement, as one Nobel laureate would do at a young age. All right, and then his educational journey. So Hamilton attended high school at the University Laboratory High School of Urbana, Illinois. He graduated from here in 3 years. He says that is due to his chemistry teachers, as they really influenced him and helped him grow into the scientist that he is today. He then went on to study mathematics at the University of Illinois for a year after graduating high school. He wasn't really super interested in mathematics, so then when he was introduced to physiology and biology, he transferred to the University of California at Berkeley in 1950. He then graduated from the University of California at Berkeley, and then later attended medical school at the Johns Hopkins University beginning in 1952, receiving his MD in 1956. And then Hamilton continued his medical training at the Henry Ford Hospital and took his postdoctoral practice to the Department of Human Genetics at the University of Michigan, Ann Arbor, where he also took his family, and they resided for, I believe, 4 or 5 years. So after that, he became an assistant professor at Johns Hopkins University, where he still resides today. And then something that he boasts about, kind of likes to talk to other scientists about, is that because it was such an influential trip for himself and as well as his family, is that he took a year to research gene sequence and arrangement in Zurich, Switzerland, actually. So he says that getting out of your comfort zone and kind of going somewhere you would not have expected yourself being in your life is really influential and can help you really grow as a scientist. All right, so he did have some career struggles. As you can see, his path wasn't exactly straightforward, as he really doubted what he wanted to do at college originally, because he initially went to study mathematics, but then he didn't really find an interest in it. So that really made him doubt what he was doing until he was influenced by biology, which is really admirable about him because he wasn't afraid to change his major into something that he really wanted to do. And it's also admirable because he is a Nobel laureate, but he didn't really know what he wanted to do originally. And that's kind of cool to think that where he is today might not have been where he is today if he had actually liked mathematics. All right, and then some of his hobbies: he enjoys spending free time with his wife, his 4 sons, and his daughter. He also enjoys listening and practicing music. So he grew up playing the piano and learning French at a young age. So he tries to contribute a lot of that to his time, or a lot of that to his life, still when he has the time, when he's not in his lab. All right, and then in 2022, he had published this paper called Fundamental Behaviors Emerge from Simulations of a Living Minimal Cell, which you can see in portion C of the diagram to your right. So their synthetic cell, JCVI, was produced as a wholly functioning cell, and then they ran simulations on this cell to see if the database called Brenda that they used, seen in portion G of the graph to your right, if that database can predict cell functions and phenotypes based on their synthetic cell here. And then some novelty about it is that this is the smallest ever procured cell as of yet, and hopefully that can allow it to become a model organism such as E. coli that can then be studied and kind of like branched off of. So they have uncovered that this synthetic cell does provide information that is applicable to other living things, and that was kind of their main goal. They wanted to see if this minimal cell that only contains 493 genes in total can allow them to compare that to other living organisms and see if, like, they will, given the same genotypes, that they will express the same functions and express the same phenotypes as this minimal cell does. All right. And then this is just my sources cited page. Thank you for watching. I hope you enjoyed.